Thanks, One Nucleus, for organising this really interesting day. When I saw the title of this session, I thought, wow, this is like the perfect fit for Alchemab, which is great. Yeah, so I'm really pleased to tell you a little bit more about how we're harnessing the human body's natural defences at Alchemab for therapeutic antibody discovery. So I wanted to start off by showing you some examples of the kind of people who really inspired us at Alchemab and whose natural defences we really want to harness. Right, so here we have an example of someone who contracted pancreatic cancer. So this is a pretty bad cancer. But 7 years following that initial diagnosis, she is still alive and doing well. And that is despite the median survival time here being around 1 year. Here we have someone in the middle who's got genetic susceptibility to Alzheimer's, but despite that genetic susceptibility, the disease doesn't progress until much later in life than would be expected. And when it does come on, the progression of the disease is very, very slow. And then finally, on the right, I think we've got someone who we all really aspire to be like. They've lived for a very long time and have had no kind of major ill health. And the question we really asked here is just why? So why are these people so resistant to these diseases? And can we then learn anything from these people that can then help us to discover drugs for the rest of us? And really, we think to understand why these people are resistant to disease, we need to look into the antibodies that they're producing. So antibodies, I'm sure you're aware of, they're produced by B cells, and typically we think of them as being the kind of one of the key players in neutralizing pathogens that infect our body. But because of the incredible diversity of different pathogens that can infect us, we have evolved this capability to produce an incredible diversity of different B cells to target all those different pathogens that can infect us. So it's a slightly unbelievable number, but any one individual in this room has the potential to produce up to 1 quintillion different antibody variants. And really, with this incredible diversity of different antibody variants that can be produced, sometimes unusual antibodies are made. So sometimes antibodies are made that don't target these foreign pathogens that infect us, but instead target our own body. They target kind of host proteins. And typically, they get a bad press. We think of them as being a bad thing and causing autoimmune disease. But I hope I can convince you that they can actually be a really good thing as well. And this is what this study on the right is showing. So I think this is a really nice example where here the authors looked at people with breast cancer, and they found that in some people with breast cancer, they could detect the presence of these naturally occurring antibodies that were targeting host proteins and giving them some protection. And those people had improved survival compared to the people who weren't producing these antibodies. So really, at Alchemab, it's exactly these kind of people who we want to find. So we look for people who are resilient to certain diseases where that resilience is driven by a B cell response. What we then do is to sequence their B cells incredibly deeply. So we're talking about millions of B cells here that we're sequencing per individual. And then what we need to do is narrow down from those millions to the specific B cells or antibodies produced by those B cells that are mediating that disease protection. Once we've found them, we then start to look at exactly what they're binding and how they function, and ultimately develop them into therapeutics. And what really kind of excited me about this approach is that it's totally target agnostic. We're not starting with a prior hypothesis about the disease and a prior kind of known drug target. We're starting by finding the antibodies that are giving these people protection, and then subsequently seeing, okay, how are they giving that protection? And through this approach, we're actually uncovering totally novel drug targets and novel disease mechanisms. Looking at the progress we've made, so a lot of time has been spent finding these people. It is not easy to actually find all of these resilient people. And we have a really high bar for what we consider to be resilience to disease. So this was really the first kind of couple of years of the company. We're now at the point where we've got over 1,500 samples across a range of therapy areas, but we're keen to be continually growing this resource. We have also developed the platform, which will pop up in a second. And I don't really have time to go into the detail of this, but it's not an easy task to narrow down from millions of antibody sequences to a handful that you think are driving protection. And effectively what we're using is a combination of artificial intelligence techniques combined with kind of good old-fashioned human intelligence to really narrow down on those antibodies of interest. And I'm happy to speak in a bit more detail about this afterwards with anyone who's interested. So then we've got the antibodies, we need to figure out what they're doing. And what we've really found here is that there's not a one-size-fits-all approach for every antibody that we found. So we've had to be developing this continually evolving suite of both techniques to figure out what these antibodies are binding, as well as techniques to figure out exactly how they're functioning. So where are we now? So since founding the company in late 2019, we're now in this position of having kind of a really nice pipeline of preclinical candidates, most of which are against pretty novel drug targets in disease. And our kind of lead one, which I'll tell you a little bit more about, is due to enter the clinic later this year. So this one that I'm going to talk a bit more about is an antibody that targets a disease called frontotemporal dementia. If you're not familiar with this disease, I think it's kind of, I mean, all dementia is bad, but I think frontotemporal dementia can be particularly bad really because of the age of onset. It has a much earlier age of onset than other dementias, and it has a relatively fast rate of progression after that onset as well. It has both genetic and sporadic causes, and actually 40% of cases are genetic. So if you kind of know that you have susceptibility to this, it's not a nice thing. And like a lot of dementias, there are no disease-modifying therapeutics currently available. So what we did was to work with a consortium called the GenFi Consortium. So this is, they have an incredible cohort of people who they have gathered who have genetic risk to frontotemporal dementia. I'm wondering whether no. You've got to really hold your nerve and not click again, even when you yeah. Okay. Yeah, so we worked with this great cohort called the GenFi cohort. So they've got people all around the world who have this genetic risk of frontotemporal dementia. And we looked at the real extremes of people in this cohort who have that genetic risk, but they've survived into old age without getting the disease. And what we found when we sequenced all their antibodies was that several of these individuals had a single antibody in common. And we started digging into this antibody in more detail, and we ultimately figured out what that antibody bound. So it bound to a membrane protein called UNC5C, which is on the surface of neurons. So that kind of made sense. It's like, oh, great, this actually, it's come from resilient people. It binds this protein on neurons. We then started to look into what that protein did. Okay, it's a survival receptor. That all makes sense. Then we looked at the genetics. Actually, there are some kind of GWAS links of this protein to neurodegeneration, yet previously totally unexplored drug target. When we tested in vivo, which you might just about be able to see, so on the left is kind of control neurons, on the right, when we put our antibodies in, you can see it kind of is stimulating some additional growth and branching of those neurons. But I think the most compelling data here is this mouse model. So we put it into a really aggressive mouse model for disease. And then we looked at the presence of NFL, which is a biomarker for neurodegeneration. So in wild-type mice at the top, they basically don't have anything. In the middle, we've got the disease model mice. They have a lot of this neurodegeneration biomarker. When we add our antibody in at the bottom, it brings that right down. And that kind of effect size that we see is really incredible for this disease model, because this is a really tough disease model. So we're super excited about this. We've got our first patient kind of dosed, hopefully this year, all going well. So really, just with that, you're going to have to invite me back next year so I can tell you the clinical data. But yeah, we're all pretty excited about that. So just to summarize what we're doing at Alchemab, we're harnessing the immune system to discover novel therapeutics. We're doing this by looking at individuals resilient to disease. It's a nice concept, but it's not been easy to execute. So there's been a lot of work building this platform to actually enable us to mine the immune system of these people and pull out the interesting antibodies. And the outcome is these novel targets we're finding, novel antibody functions, really interesting mechanisms of action of some of these antibodies as well that you wouldn't typically be able to easily engineer for. Oh, and then just finally, I'll let it come up, but just to thank kind of everyone who's been involved with this. We've got a lovely team at Alchemab. We started off being based in Babraham, now based down in Whittlesford, but still have very strong links to companies here at the campus. Thank you.